Hey y'all, back by popular demand, I have decided to make a new video for you all regarding exam 4, right? So let's just get right into it. This one should be relatively short. We're just going to go over the three main topics being DNA replication, the cell cycle, and mitosis meiosis. So, let's just start generally with the cell cycle, right? Well, let's start a little smaller. So here in the cell cycle, we have four phases, and it goes this way. We have G1, oops, S, G2, M, right? Just like that. G1, S, G2, M. So in this case, G1 stands for growth, or just the G in general stands for growth, and it stands for gap, right? So it's a gap between the previous mitosis and the S, and G2 is a gap between synthesis and mitosis, right? So S stands for synthesis, of course. So what are we synthesizing? DNA. And M phase is, of course, mitosis. So yeah, real simple. This is your cell cycle. And one more thing. If we kind of cut off here, just like that, right? Kind of like that. The space between G1 through to D2 is called interphase. It's called interphase, right? So interphase is pretty cool. Interphase, you do all your growth, all of your normal activities, all the metabolism, everything. Everything's happening during interphase. Just normal cell activity, right? What doesn't happen during interphase, however, is cytokinesis. Cytokinesis happens right here. It's technically not part of mitosis. That's cytokinesis, right? So cytokinesis is not included here. So you may see something like that, right? So good. Let's go a little bit more in depth what happens during S phase. So what happens during S phase? So S phase, of course, is synthesis phase. So what are we synthesizing? We're synthesizing DNA. And how do we synthesize DNA? Well, let me show you. So, you start with a strand of DNA. Geez, that was really bad, huh? So we start with a strand of DNA, and normally DNA, you can't really access it because, of course, it does have a double helix, right? So how do we go ahead and access this DNA? What can we do to break through? Well, we need the help of an enzyme. An enzyme known as helicase. So I'm gonna write out the few enzymes that you do need to know. Helicase, DNA polymerase, R RNA primase, or just, in other words, just primase. Usually just primase, and ligase. Normally there would be five. The last one is topoisomerase. I think it'd be important to know what it is. I don't think you should consider yourself really. I don't think you should really concern yourself with what it does. Right. So here in BIOL-2101, we like to simplify our DNA, how we make DNA here, right? So, just real simple. That's one strand of DNA, that's another strand of DNA. So we're unzipping the, one sec, so let me draw in some bases. We're unzipping the helicase, right? So I like to draw the helicase as a triangle. So yeah, the helicase is what's unzipping the DNA, right? So remember, we have the DNA that kind of looks like this, right? So we just have to cut down the middle. We're unzipping it, therefore, right? Just like a zipper. So that's what the helicase does. It just goes on and breaks those hydrogen bonds that maintain the structure of the DNA, and it just comes in and breaks it in half. So what happens next, right? Well, we have to initiate DNA replication somehow, and we can't just jump into a DNA polymerase. Just we can't just jump into it right away, right? What we have to do is we have to start with something known as a primer. Or so primers are made of RNA, right? So what do we do? What do we do with a primer, right? So what we do is we go here onto the DNA. We have a special enzyme known as RNA primase. That's RNA primase, right? And what RNA primase does is that it goes in and it adds. Okay, well, let's see what I can do here. Oh. Maybe not. Okay. Primase just goes in here and adds a primer. So I'm going to draw a primer as red, right? Just kind of like that. And usually these primers are like 10 base pairs. I'm simplifying it a little bit, just showing you 2. Right? So we have to start with a primer, right? And now we get into the polarity of the DNA, right? And what I mean when I say that is we get into which side is 3' and which side is 5'. Right? So here we have the 3' side and the 5' side, right? That's wrong. Sorry. We have the 5' side here and then the 3' side here, right? And then the 3' side here and the 5' here, right? So the thing about this, why we draw these 5', 3', what have you, the reason why we do this is because DNA runs anti-parallel, right? So we have one going this way, one going this way, right? So DNA is going, 2 strands of DNA are going opposite of each other. So why is this of concern to us, right? Because, and this is important to note, DNA is always, I'm going to underline and bold it just to show you how important this is. Always synthesize. Ugh, come on. From 5' to 3'. Right? DNA is always going to be synthesized from 5' to 3'. So, what does that mean? It means that we have another special enzyme known as DNA polymerase. So here we have the RNA primer. So we have the primer made of RNA, right? That's our primer. It's in red. So the polymerase, the DNA polymerase comes in, it recognizes this primer, it says, okay, that's where I need to bind. And then it goes and synthesizes DNA from 5 to 3, right? So in this case, it just kind of goes this way, right? And let's see if I could do a decent job of drawing this or depicting this. Kind of goes that way and then makes DNA. It adds nucleotides to the already existing sequence, right? Just like that, right? So we have 5 on this strand. We have 5' to 3'. So on the newly made strand, it's going to be opposite. So on this side we're 5', and on this side we're 3'. So this follows the same rule. We're going from 5' to 3', and we're moving inside the replication fork, right? So that's on this side. We're moving inside the replication fork. Right? When the 3' is facing inward, we know it's going to replicate faster, and it's going to be the leading strand. right? So we have two strands, the leading strand up here and the lagging strand down here, therefore, since we already figured out what the leading strand is. So why do we call it a leading strand and a lagging strand, right? Because down here, the DNA is going to run the opposite way, right? Kind of like this. So what does that mean? It means that because we're moving the opposite direction, we have to have a primer. So remember the primase. We have to have a primer here, and then the DNA polymerase comes in, attaches here, and then it goes this way. All right, so we make more that way, and then more of the DNA is opening up, so we have to do it again. There's a primer here, and then RNA primase attaches here. Sorry, no, that was wrong. No, RNA primase makes the primer here, and then DNA polymerase attaches here and makes more of the nucleotides, just like that. Right, so the thing about this is that we have to remove these red bits here, right? We have to remove the RNA. So it's not really a detail that you need to know, but it's another RNA polymerase that comes in and turns these white. Oh, that's not what I wanted. Okay. Kind of like that, right? Except the thing is that in its wake, it kind of has like a little gap that's formed, right? So we want to seal those nicks. We want to make sure that these gaps no longer are present and that the DNA is continuously flowing, right? And we do that with another special enzyme known as ligase, right? So ligase comes in, essentially like glues together these two. It essentially glues together the DNA together. So it just kind of does that, a continuous strand. The ligase is yellow, so it's right here. So it comes in, glues this together, and we're all good. So kind of looks like that, right? When we're talking about the lagging strand, right? So just to be clear, we have the leading strand here, and over here we have the lagging strand. Right, so we kind of talked about, you know, how the fact that we have multiple fragments of DNA, and how the more the DNA is unzipped, the more primers we have to have, right? So when we have these primers, the fragments of DNA that exist between them are called Okazaki fragments, named after the dude who discovered them a while back. So these are called Okazaki fragments. Right? So, just to review, we have the helicase, which unzips the DNA, the RNA primase, which adds a primer, which is a signal for the DNA polymerase to attach, and the DNA polymerase is the one doing all the polymerizing. It's making the DNA, it's just going, and then the ligase, and then the DNA polymerase goes ahead and removes these primers, and then the ligase comes in and seals the gaps between the strands that were made. The only one that we didn't discuss is the topoisomerase, which is basically just like a protein that exists over here, right? So this is after the helicase, or before, rather. So this is before the helicase. And it just relieves tension for the DNA, so to prevent supercoiling. So yeah, it's not super important that you know the topoisomerase, but yeah. So cool. Another thing is that this is important, right? When we're moving inside the replication fork, when the arrow is facing towards the replication fork, and just to be clear, I mean, if you look at a normal fork, right? So this is a fork. So the place where like everything kind of sticks out, that's like the tail end of the fork, right? Or the head of the fork, I don't even. Here is your fork. This is your replication fork, right? this region right here. So if we're going inside the replication fork, that is the leading strand. If we're going outside the replication fork, that is the lagging strand. So good. So all of this happens during S phase, right? Which we discussed. All right, so we discussed S phase. We discussed all of that. So good. So now what we're gonna do is we're gonna go over the difference between chromosomes, chromatids, homologous, all of that sort of stuff, right? So normally, a cell kind of looks like a bowl of spaghetti, right? But for now, I'm going to simplify it a little bit. So let's say we have a cell of 2n equals 4. So what does this number tell us? 2n equals 4. Well, for one, it tells us that this is a diploid cell. And what does it mean for something to be diploid? Well, a diploid has or contains 2 sets of chromosomes. Right? And this number tells us the number of chromosomes. So 2n equals 4 tells us it's a diploid cell with 2 sets of chromosomes and has 4 chromosomes in total. So therefore it has 2 sets of 2. Humans have 2n equals 46. So therefore humans are diploid, contain 2 sets of chromosomes with 46 total chromosomes, and has 23 pairs of 2. So what does this mean? Right? So when we say we have 2 sets of chromosomes, we really mean is that homologous chromosomes are present. So let's go ahead and divide these off a little bit. Yeah. So when something contains 2 sets of chromosomes, it means we have homologous. So homo, right, means same. So homologous chromosomes, that's the same thing. It means it means the chromosomes they have the same genes, they have the same length. They do not have the same nucleotide sequence. Yes, all of those good stuff. Right? So this is a cell in G1. So let's go ahead and analyze this. So this is one set of chromosomes, and this is the other set of chromosomes. So now, I want you to ask yourself, are there any chromatids pictured here? And the answer to that is no, there are not. We do not have sister chromatids until after S phase, right? So after the DNA has been synthesized, the DNA has been duplicated, right? You've made another batch of DNA, right? You've made an exact replica of all the DNA. Only then do we have sister chromatids. But here during G1, we do not, right? So how many chromosomes do we have? Well, we have 1, 2, 3, and 4. Okay, good. Now, why is one pair bigger than the other pair? Well, we say that we notice this, right? Because these are your homologous pairs, right? So when we say 2 sets of chromosomes, this is kind of what we mean, right? So let's color these actually. And this is going to really come in handy when we talk about meiosis. Whoops. So notice how one pair is notice how some of them are red and some of them are blue, right? So you get red from mom, for example, and blue from dad. So look here, right? This is one pair, and this is another pair, right? So this set of chromosomes, the blue set, you get from your dad, and the red set you get from your mom, right? So why this is important is because later on we're going to see the genetic variation that comes into play and how mom's chromosomes can go into one cell and dad's can go into another, right? But we notice these lengths are similar, right? So both of these chromosomes contain the same genes, just like this one. These contain the same genes. There's of similar length. So let's see. Let's write it out. They are of similar length, contain the same genes, but do not have the same nucleotide sequence. So I always like to pull out the example of Harry Potter, right? If you ever read the first couple of Harry Potters, they're around like 400 pages ish, right? So they're set in the same universe. There's part of the same book series, right? They have similar content in them. They're about the same length. However, they do not have the same content within them. They're different stories, right? So if you have the first one and the second one, they're different stories, right? Just like how with this pair, they contain the same genes, they are similar length, but they do not have the same nucleotide sequence, right? So just I know this is confusing. I know. Um, this these red ones you get from mom, and these blue ones you get from dad, right? It's not like you get both of these from mom and then both of these from dad. No, from in each pair you get one from dad and then one from mom, right? So homologous chromosomes are always going to be in pairs, right? Because they are having the similar length and similar content, right? So, yeah, homologous chromosomes. These are your homologous chromosomes right here. Good. So, once we go through S phase, now let's draw our cell. So now, our cell looks something like, our chromosomes look something like this. And for posterity's sake, let's go ahead and color these ones as well. Right? So now, what is different? Now the DNA has been duplicated, it's been replicated. There's twice the amount of DNA during G2 phase as there was in G1. However, right, and this is where it gets confusing, so pay attention, right? This is one chromosome. This is also one chromosome. This is one chromosome, and this is one chromosome. So we are still 2n equals 4. Now the only difference is, and this is the only difference, is that the DNA has now been duplicated, right? So within each one chromosome, we have two constituent sister chromatids. Just like that. We have two sister chromatids in every one chromosome. We do not have sister chromatids here because the DNA has not been duplicated. Here after S phase, we do have sister chromatids, right? So the thing about sister chromatids is that when they're sisters, they are exactly the same, right? So it's not like over here where it's not like with the homologous chromosomes where they're a little bit different, but they're kind of the same. No, sister chromatids are exactly the same. So here in this cell, now we have 8 chromatids, 4 chromosomes, and 2 pairs of homologous chromosomes. 2 pairs of 2 homologous chromosomes to be specific. Right? So this is still a homologous pair, and this is still a homologous pair. Right? No, that didn't change from over here. We still have the homologous pairs because we still have 4 chromosomes. Right? Just now we have sister chromatids, and during mitosis these sister chromatids are going to be split apart, right? So good. I think it's very important to make that distinction between the sister chromatids and the homologous chromosomes, because it seems like nearly everyone likes to or doesn't like to, but seems to tend to get it wrong. So with that all being said, let's go ahead and move on to the actual mitosis. Right, so we just discussed in G2 how we have sister chromatids, right? So let's have a cell here in the corner. This is our cell 2n equals 4, right? So there was a signal from outside that tells the cell to, okay, you're clear to divide. So now what happens is we go into prophase. Right? So what happens during prophase is we have the cell, right? And we have the DNA over here, which looks all like condensed, and it doesn't look all condensed. It looks all messy and like a bowl of spaghetti. So during prophase here, now we have our chromosomes that are visible, right? And then we also have the centrosomes that move to each pole. So the centrosomes, we'll get to them in a second. So that's what happens during prophase. And then we get to the one that everyone forgets about, prometaphase. So now we have prometaphase, right? So now during prometaphase, the cell is going to go ahead and disintegrate the membrane, because to get to the DNA, we have to go ahead and disintegrate this membrane, right? So now the centrosomes are going to attach over here, just like that. Yeah, just like that, right? So a few notes, right? These yellow things are centrosomes. And let's have a little look-see. That's a chromosome, right? And at the intersection point, that's called a centromere. So a centromere is the intersection point between the chromosomes. A centrosome is an organelle that has spindle fibers. So these are your spindle fibers right here. That has spindle fibers originating from it and attaches at the centromere of the chromosome. So these spindle fibers, right, they originate from the centrosome, and they are made of microtubules. Made of microtubules, right? So make a note of that. The spindles are made of microtubules, and they originate from the centrosome, right? So that's what happens during prometaphase. And then metaphase, it's all pretty straightforward from here, or at least it's straightforward to me because I've been teaching this for a little over a year now. So now these chromosomes are going to align at the middle. These spindles are going to push them here towards the middle, right? Kind of like that. So that's metaphase. So the point of metaphase is just to have a clear idea. So the cell has a clear idea. Okay, all these chromosomes are accounted for. they're all in the same position, we could have this. We could now have them split apart perfectly. So now I want you to remember this mental image. This right here. Remember what metaphase is. Remember what this looks like. Do not forget it. So now we're going to anaphase. Right, so anaphase. So we have the centrosomes. And then it kind of looks like that. So during anaphase, the chromatids are being pulled apart. That's important to note. The chromatids are being pulled apart, right? Because remember, the chromatids are exact replicas of each other. They are the exact same. So once we split the cell in half, we're going to end up with two identical cells, right? So this is anaphase, right? And then after anaphase, we go to telophase. This is telophase, right? And during telophase, we have what's known as a cleavage furrow that forms. So this like little sort of butt-looking thing, that's a cleavage furrow. And now we undo everything we did during prophase and prometaphase. We start to reform this nuclear membrane, and the chromosomes start to decondense. Just like that. Right? And then, after all of this, we go through a process called cytokinesis, which was just the splitting of the cells. And then we end up with two. Look at that circle. That's really good. We end up with two circles. Sorry. We end up with two cells that are pretty much identical, have the same amount of genes, the same amount of chromosomes, and now they're pretty much exactly the same as what we started with. Right? So these are both 2n equals 4. So that was mitosis, right? So we have prophase into prometaphase into metaphase into anaphase into telophase, and finally cytokinesis. We make two cells, right? Prophase condenses the DNA. Centrosomes, prometaphase destroy the nuclear membrane. Centrosomes attach via spindles that are made of microtubules. Metaphase, everything's lined up, all the chromosomes. Anaphase, everything's split apart, all the sister chromatids are split apart. So after anaphase, right, here in each cell we have 4 chromosomes, right? Telophase, we undo what we did in prophase and prometaphase, which means we decondense the DNA and we reform the nuclear membrane, and yeah, we want 2 cells. Good. So now let's go over meiosis. So meiosis is a fun little thing, right? So we were talking about the cell cycle earlier. Meiosis is a sort of deviation from the cell cycle, right? So if you imagine the cell cycle, how we drew it, you go from G2 to M phase. So now what's going to happen is it's actually going to kind of shoot off, and it's going to go from G2 into the meiosis phase. And this happens from a special type of cell known as a germ cell. So good. So here's what happens during that. So we have 2 meioses. I don't know if that's a word, but we have 2 stages of meiosis. Meiosis I and meiosis II. So let's talk about the first one, right? So we're just going to start... I just want to go ahead and copy this because I'm lazy. So we start off with the same cell, right? 2n equals 4, nothing special here. So now there's a special signal that comes in and says, okay, you got to start making sex cells. So this cell is now going to turn into a gamete. A gamete is just another word for a sex cell. So you have gametes and you have somatic cells. Gametes are sex cells. Somatic cells are just normal body cells, meaning your lymphocytes, your adipocytes, your skin cells, your brain cells, all of those. So let's go into prophase 1. So now there is a really important distinction to make between mother's DNA and father's DNA. So now we're going to see that in full play. So prophase 1 right? We're going to draw. We don't have a prometaphase 1 or a prometaphase 2. We just do everything during prophase. So we're going to go ahead and disintegrate the cell membrane, the nuclear membrane. Nothing special there. We're going to have our centrosomes at each pole. Once again, nothing special there. But here's what's going to happen now. We have mom's chromosomes and then dad's chromosomes, and these are going to intersect like this. Kind of like that, right? We could draw it a little better here. Yeah, that's good. So these are going to intersect, right? And this intersection point there's something special happening, and that's called crossing over. So at the intersection point, we have crossing over. What is crossing over? It is the exchange of genetic information, right? So remember, homologous chromosomes are of similar content, similar length, but ultimately they're different, right? So these chromosomes are actually going to exchange information. They're going to exchange genes. And this is a form of genetic variation, right? So a child made from this gamete way down the line is going to be much more randomized than something like, I don't know, like a bacteria would be. So yeah, I hope that's clear. So crossing over happens here. And yeah, that there's a bit. So this already there's a pretty big deviation from just normal mitosis, right? So now moving into metaphase 1, we'd have a couple different things happen here, and you're going to see the difference. You're going to see the difference, right? Oh, I forgot one thing. During prophase 1, same thing happens. We have the spindle fibers that attach at the centromeres, right? Same thing. So, now during metaphase 1, here's another big deviation from normal mitosis, right? Look at what happens here. So the spindle fibers move it to the middle, just like this, right? So notice how they're still attached, right? But also notice, right, if we split it in half preemptively, if we kind of look like that, one daughter cell is going to end up with all of mom's chromosomes, and one daughter cell is going to end up with all of dad's chromosomes. This can change. So there's another way we could actually draw this. Look at this. Oh, whoops. Okay. I could do that a little better, but that's okay. So now look at this. Look how these are different, right? If you cut it in half, this way, now, whoops, come on. Now one cell has some of mom's, some of dad's. The other cell has some of dad's, some of mom's, right? Versus this one has all of mom's, all of dad's. The other one has sorry, one has all of moms, the other one has all of dads. Right? So this process is known as independent assortment. If I could spell, Jesus. This process is called independent assortment, where the chromosomes are going to assort each other independently of each other, right? So we could have, so it's not always going to be set in stone. Like mom's is going to be in this gamete, dad's is going to be in this one. No, that's not how it works. They're going to randomly assort themselves. And this has to do with more genetic variation, just like crossing over. So I'm gonna go ahead and erase this, just to make it a little less cluttered. So now we go into anaphase 1. So now what happens is normally, right? Normally, the sister chromatids would be split apart, but now, here's a big difference. Now, the homologous chromosomes are being split apart. And we're going to see why that's important here in a second, right? Now the homologous chromosomes are going to split apart. So we go, like, once again, we go down the middle, we can see that one cell's going to get moms, one cell's going to get dads. That's just for simplicity's sake, right? And also, I also want to show you this, right? So some of these chromosomes kind of gonna look like that, right? Where you have bits of red in the blue chromosomes and bits of blue in the red chromosomes. That's just a result of crossing over. I won't draw the rest of the chromosomes like this, just for my sake, but just know that's kind of what they look like. These are all results of independent assortment. No, sorry, these are all results of crossing over. So good. Let's now move into telophase 1. So now, I just realized I can't really do that. So now we have the cleavage furrow forming. We have the nuclear membrane reforming. We have the DNA decondensing. And that's it. So, pretty ordinary telophase, if I say so myself. This is telophase 1. Throughout all these processes, the telophase normally stays the same for the most part, right? And I just realized I should probably draw it kind of like that, right? So good. So now we make 2 daughter cells. So we reform the nuclear membrane. Our DNA is decondensed. Kind of like that, right? So notice, this is really important. We still have sister chromatids. So I hear you misconcepting. I don't think it's a word. If they have sister chromatids, is the cell number still 2n equals 4? Is the chromosome number still 2n equals 4? To which I answer to you, no, it is not. Right? So during metaphase and during anaphase, we're splitting apart homologous chromosomes. So homologous chromosomes, despite being similar, are still the same chromosomes. What? Don't ignore what I just said. Homologous chromosomes, despite being similar, are still both chromosomes. They still both count as their own chromosomes. Chromatids do not count as their own chromosomes until they are split apart, right? So now, since the homologous chromosomes no longer exist, after telophase 1 and after meiosis 1, since they no longer exist, these cells are now n equals 2. Let me make that white. These cells are now both n equals 2 because we split everything in half, right? We split the chromosome number in half. We no longer have homologous chromosomes. So excuse me, we no longer have homologous chromosomes, so the cell is going to just have half the amount of what we started with. So not 2n, but n equals 2. So when we say n equals, n tells us that this is haploid. And 2 tells us the amount of chromosomes. So haploid tells us that there's only one set of chromosomes, and there's only one set of chromosomes, which therefore means there are no homologous chromosomes. Good. So let's go ahead and run with this cell, the one with all of mom's genes. So now we're going to discuss meiosis II. Right? So meiosis II is pretty much exactly the same as mitosis. Right? And you'll see what I mean when I start doing it. Right? So we draw the cell, right? The nuclear membrane starts to deform once again. The chromosomes condense, and we're going to have one big and one small. See, notice how these are not homologous. They are vastly different sizes. We have 4 chromatids, 2 chromosomes, no homologous chromosomes. So yeah, same thing, same ordeal. We have the centrosomes here at both ends, attaching the spindles at the centromere, just like that, right? So that's prophase 1. Sorry, ignore that. That's prophase 2. Right? So, let's go into metaphase 2. Metaphase 2 is pretty much exactly how you might expect, right? So we have the centrosomes here at both poles. We have the chromosomes lining up in the middle. And we have the spindle fibers orchestrating all of this, right? So this is metaphase 2. So, moving into anaphase 2, now, remember, in anaphase 1, the homologous chromosomes were split apart. Now, during anaphase 2, the sister chromatids are going to be split apart. Just like in mitosis. Just like that, right? Pretty ordinary. This is anaphase 2. Right? And then telophase 2, I mean, what more is there to say? The cleavage furrow forms. The nuclear membrane is reforming. And the DNA is decondensing. So now we have 2 daughter cells. But remember, we had another cell, didn't we? We did. So we're going to do this exact same process all over. Just this time, it's going to be for the paternal, the other cell. So I'm not going to draw all of this out again, but I will say this. We have 2 cells from this one that form then now it'll kind of look like this. Right? So both of these, or sorry, no, all four of these are n equals 2. Because we have one chromosome, two chromosomes. One chromosome, two chromosomes. We don't have any chromatids because we split apart the chromatids. Remember? We don't have any homologous chromosomes because we split that in anaphase 1. So we have n equals 2, meaning that it's haploid. Right, so here's a couple of fun facts about haploids. And they're not fun because these are facts you have to know for your exam. So write these down. Haploid cells can undergo mitosis, cannot undergo meiosis. So haploid cells can undergo mitosis, right? So you see that with mushrooms. Mushrooms are an example of this. Mushroom cells, humans kind of exist as diploid cells. Mushroom cells exist as haploid cells, right? So mushrooms, they can grow, they can expand, they can make more of themselves, but so that means the haploid cells can indeed undergo mitosis. However, haploid cells cannot undergo meiosis, right? Because remember what we did during meiosis? We split the genome in half. We split everything in half. We can't do that again. It will be like 1/2 equals 1 in this case. That doesn't make any sense. So yeah, very good. I would ask you if you had any questions, but obviously I can't really ask you that. Um, I'm just gonna go over what else you might need to know. Um, oh, well, it kind of goes without saying that you kind of need to know the what? Whoops. It kind of goes without saying that you need to know the purpose of all of this, right? So the purpose of mitosis is for an organism to grow, survive, and prosper. So I'm sure you were all children at one point. I mean, I'd be really surprised if you weren't. But we all have to grow, right? We all have to grow. So you were a kid once, but how did you grow? You drank your milk and ate your veggies? Well, no. Well, I'm sure that has something to do with it, but in order for you to have grown, your cells had to undergo mitosis in order to make more of themselves, in order to make more mass, and to make yourself bigger, right? So mitosis is for an organism to grow, survive, and prosper. Now, on the other hand, meiosis is solely for the express case of sexual reproduction. Because meiosis produces sex cells, right? So if we want to make sex cells, then what's the point of sex cells? To make more organisms, right? So when a sperm and an egg come together, they make a baby. Well, that kind of goes without saying. If you don't know that, then I'm sorry. I feel bad for you. Sorry. So meiosis is for the express and sole purpose of sexual reproduction. We don't make we don't under no all of our normal cells don't undergo meiosis. That would be kind of chaotic. That only applies to our sex cells. So good. What else do you need to know? So we discussed when the cells become haploid in meiosis. That's after meiosis I, right? We talked about that. So we talked about DNA replication. DNA replication does not occur between meiosis I and meiosis II. It only occurs during S phase, and like I said, it's like a deviation, right? So yeah, I think I've covered just about everything. So yeah, that was if I were you guys, um, I would be very thankful because this exam is. If you've been paying attention in class and if you've taken notes during this video, this exam should not be very difficult. In fact, this exam, I would have been jealous if I was in your position because when I had to take this exam, it was much harder. So, I implore you, watch this video as many times as you need. I cover pretty much everything that you need to know for your exam. So, yeah, with that in mind, I hope you all have a good night or a good morning, depending on when you watch this. And I'll see you all tomorrow, probably. Maybe not all of you, but in any case, take care. Have a good night.